Okay. Thank you folks for being here. It's 4:10, so we'll go ahead and get started. It's my pleasure to introduce Natasha Dhamrait. She's the resident lab artist and master gardener, and also does some research we'll hear about. She came to the lab, it's her third year in the lab from the University of Toronto. Excited to work on transposable elements, which we'll hear about today. So picking a really easy topic to start off with. And then she chose a really easy genome like maize to work in. And although we have lots of high quality assemblies for maize, where we have full length transposable elements assembled and you can like just count them, that was also too easy. So Natasha has chosen to work with short read data in wild populations and heterozygous material. And although I don't think she'll be talking about it today, spent a huge amount of time in her first year or so here, trying out lots of other methods to deal with these data, and finding out that most of them are really terrible, and spending a lot of time showing how bad they are, and has instead come up with a really clever approach for using short read data to think about evolution and population genetics of transposable elements in wild and cultivated populations. So that's the work she'll talk about today. Good answer.
Thank you everyone for coming. Today I'll be presenting my research that I've done over the past couple of years, the research I plan on doing for my remaining years here. My work, as Jeff mentioned, generally revolves around the intersection of transposable elements and the evolution of maize, more commonly known as corn, and its wild relatives, the teosinte. Starting, we're going to pass around some cobs. Each has a short description. Please be gentle with them and treat them as the precious but delicate specimens that they are. And once they've gone around the room, if you can just pass them over back this way, that'd be great. And if at any point I'm talking too quietly, as I have a tendency to do, please just let me know. Thank you. So to start off, maize is culturally, historically, and spiritually important to billions of people throughout history, especially in the Americas, where it was domesticated. I do believe it's important to mention that as a maize genetics researcher, my family has no geographical, cultural, or social ties to maize. My appreciation, my interest, my perspective in maize comes from a personal and academic one rather than an intergenerational one. It's additionally important to acknowledge the land we're on is not our own. It is the land of the Miwok and Patwin people, and I have lived, learned science, and done research on the lands of the Ohlone, Muwekma, Post Miwok, Anishinaabewake, and Haudenosaunee people, and the Mississaugas of the Credit First Nation. The evolution of maize and its wild relatives is intricately linked to people, and it's only when recognizing both the human history and the maize history that we can start to understand biological phenomena in this genus. And with that, let's dive in. So I'm going to start with some background information on the history of maize and its relatives, and then I'll get into my own research. So the teosinte are native to Mexico and parts of Central America, and they span a wide growing range from arid subalpine pockets of the highlands of Mexico to warm, wet, tropical forests of central Mexico. These diverse environments and ecologies have created diverse populations despite hybridization in range overlaps. I specifically point out these three teosintes on the map, Diploprennis, Parviglumis, and Mexicana, because these are the three teosintes I focus on in addition to maize. Phenotypically, the three teosintes, Mexicana, Parviglumis, and Diploprennis, don't differ greatly. You look at them and think, yeah, that's a grass. They're generally branching with tassels at the end of stalks, and they have large leaves extending from the stem. And the kernels, the seeds of the teosintes, also all look pretty similar, kind of like a triangular prism encased in a hard fruit case, where the fleshy part of the seed is inside that fruit case. And I'm sure, as you've noticed, none of these seeds necessarily look like corn. And then really fast, I'd like to point out the divergence times for these three teosintes are on the lower side for plants. The split between Parviglumis and Mexicana was only about 30,000 years ago. So, and there's been consistent gene flow between all the subspecies since then. This is plenty of time for selection and divergence, but not enough for vast genomic differences, which presents a rare opportunity. This genus is a fun opportunity to study how different environments and different life histories can shape the genetics and genomics of close relatives. But how do we get from teosinte to maize? The answer is domestication. Most of the foods that we eat have been altered by humans over thousands of years through a continual evolutionary process called domestication. Domestication reshapes genomes, radically shifts population dynamics, and alters selection pressures on large swaths of the genome. Maize, Zea mays maize, was domesticated by indigenous peoples of the Americas at least 9,000 years ago in the Balsas River Valley of Mexico, from Zea mays subspecies Parviglumis and mexicana. The process of domestication remodeled teosinte, the recalcitrant weedy plant with hard shells, to the edible, nutritious, and hardy crop that we now know as maize. But why should we, as population biology enthusiasts, care about domestication and study it? That's because domestication, we believe, is an ideal model to study adaptation, selection, and genome evolution. Due to the concise timeline of domestication, many signatures of selection are still detectable in maize and the teosintes. Additionally, the presence of wild relatives, like teosinte, allow for useful comparisons between domesticated and non-domesticated sister taxa to inform evolution and adaptation. The timeline and the intensity of selection pressures associated with domestication fall well within the range that we observe in natural populations, allowing for patterns observed in domesticated taxa to inform the study in natural systems. And as you can see in this figure, there are obviously some phenotypic differences between teosinte and maize. Many of these trait changes were actually caused by TEs or linked to a TE insertion, such as reduced branching in maize so that more plants can be packed tightly in a single field, the removal of the hard fruit case so that the fleshy kernel is more accessible, and the reduction in the number of ears per plant from many to only one or two that we see in maize. Early work in maize domestication relied on inferences primarily made through morphological, cytological, and archaeological evidence. The oldest archaeological evidence we have of domesticated maize are around 8,000-year-old microfossils. And the earliest archaeological evidence we have of whole maize cobs are several thousand years younger than that. Morphologically, as you can see in that top figure there, a lot of these preserved maize cobs more closely resemble maize than they do the teosintes, implying that domestication could have begun many hundreds to thousands of years prior than what we expect. Starting in the genomics era, a lot of domestication work switched from examining patterns of how rapid and intense selection during domestication restructured genomes and the potential for improving breeding. However, our understanding of early domestication and the geography of that early domestication is still out of date given our current genomic resources. Part of the reason why genomic work in maize is a little bit tricky is that maize is incredibly diverse. Just look at some of the cobs that are being passed around the room. It's hard to imagine that all of these came from the same species. Indigenous peoples of the Americas have selected maize to have different flavors, colors, hardiness, tolerance to various abiotic factors, resistance to disease, depending on the wants and needs of their communities. The diverse selection pressures on maize have created a diverse genome with many adaptations, some of which are shared and some of which are unique between populations. Now, people around the globe grow different maize varieties for countless purposes. Indigenous farmers and agricultural breeders continue to shape the diversity of maize through adaptation, seed exchange, breeding, but there's still little acknowledgement for the grain that provides more than 60% of the carbon in our bodies, and very little acknowledgement for the people whose knowledge, innovation, and seed feed the world today. In addition to feeding the world, maize has also shaped the field of genetics. It's been a model organism in the field for decades, and many key findings have actually come from maize, including the first evidence of crossing over during recombination. This was discovered by two icons, Harriet Creedon and Barbara McClintock in 1931. Fast forward several decades, we now have many genomic resources, which Jeff alluded to, and many fascinating breeding innovations, including data on long-term selection breeding projects, large mapping populations, and many fully assembled reference quality genomes. The wealth of resources in maize allows us researchers to validate and explore inferences about biological phenomena from many different directions. But I think the most interesting thing to come out of maize genetics research is transposable elements, or as I'm going to refer to them in the rest of this presentation as TEs. TEs are genetic elements that can copy themselves and move to new positions within the genome. They were discovered 70 years ago by Dr. Barbara McClintock, and her discovery eventually won her a Nobel Prize in 1981. These images on the bottom are a few charismatic examples of TE-induced phenotype changes in crops, including the elongated form of Roma tomatoes, Mendel's wrinkly peas, color variation in grapefruit, and I have several examples in maize being tested on brim. We now know that the vast majority of plant genetic material is derived from TEs, and TEs are found across the tree of life. However, TEs are not a monolith. Not every TE is the same. TEs, just like organisms, can be grouped into taxa by phylogenetic relatedness and similarity in various characteristics. This figure shows a phylogeny of TE superfamilies. Within each superfamily is many TE families, and within each TE family is many TE species. I don't have divergence times on this phylogeny, but it's theorized that the split between class 1 and class 2 TE superfamilies actually predates the split between eukaryotes and prokaryotes. Even closely related TEs within the same superfamily or within the same class can be millions of years diverged, such as Ty1 and Ty3 TE superfamilies. These superfamilies diverged around the same time as palm trees and giant squids. Yet most geneticists and genomicists consider these two superfamilies to do the same thing in the genome and experience the same evolutionary forces. But for these two TE families specifically, how do they differ in a single genome, like the maize genome? When comparing Ty1 and Ty3 superfamilies, we see that even within and between these superfamilies, there's variation in the average length of TEs, how far from genes those TEs are located, and how often TEs in the superfamily insert in other TEs. And this variation exists when we look at all superfamilies in the maize genome for several different characteristics. Someone could spend their entire PhD exploring this variation. But let's focus on just the first panel, which is TE length. I find it particularly striking how variable TE superfamilies are in length. Some of them are only a few hundred base pairs long, where others are several thousand base pairs long. My key point here is that TEs, even within a single genome, even within a single superfamily, are incredibly variable in what they're doing, how they're doing it, and where they're located. So given that TEs vary within a genome, how do they vary between genomes? In maize and the teosynthase, TEs are incredibly polymorphic. Let's walk through one example. This is the bronze locus in the maize genome. We see for this single genome, there are some TEs, some are inserted into others. Well, let's look at two genomes. We see there's different TEs inserted in different locations. And for 6 genomes, we see these genomes differ in the number of TEs inserted, how nested those TEs are, and where those TEs are located and what TEs are present. This level of variation exists in gradients throughout the genomes in this genus Zea. Across only 4 varieties of maize, more than 7,500 TE families were found in only a single genome, and about 20% of the genome was variable across those 4 lines. To put those numbers in reference, in perspective, that means that each maize genome, on average, has the equivalent of 2 Arabidopsis genomes worth of unique TEs. 2 Arabidopsis genomes worth of unique TEs. And while we know this variation exists, between species and the subspecies in this genus, what drives that variation, and how it differs on the population scale is still unknown. And we've talked a lot about TE variation, but one very important thing that most TEs have in common is that they're generally deleterious, which makes sense. Think about a 1 kilobase chunk of DNA inserting in a gene. That's probably going to impair function. TEs also do a whole host of other things that could impact fitness and generally destabilize genomes. And while it's true that some TEs can be and have been adaptive, I want you to generally consider them deleterious or neutral at best. To put numbers to this assumption, for recent work has shown that on average, a single maize plant loses 1 kernel of yield for each additional 14 megabases of TE content. When you have 80% of your genome as TEs, and I don't know how many hectares of maize are grown throughout the world, that's a significant reduction in fitness directly due to TE content. As a result of the continual TE activity over evolutionary time, genomes have developed several mechanisms for controlling TEs. In plants, this is mainly through epigenetic silencing and chromatin remodeling or methylation. General patterns of TEs, activity, dynamics, adaptive potential, etc., are hard to generalize due to the diversity of genomes and the diversity of TEs, but also because TEs can interact. TEs can compete for space, they can parasitize each other's replication machinery, they can even facilitate closely related TEs. This figure shows changing frequencies of 2 TE species, Grande and Doke. Doke replicates by inserting into Grande, which means Grande can no longer function. This means as Doke increases, Grande also increases until we see predator-prey bust-boom cycles that we often associate with species. TEs evolve with genomes, and genomes evolve with TEs, and TEs evolve with TEs. And due to the close relationship between TEs and genome function, modern TE abundance is a direct result of past evolutionary history, and thus can inform past evolution. All of this leads me to my research. For my dissertation, I'll be researching how TE family abundance and diversity varies on different scales and what drives that variation. For chapter 1, I'll explore how the diversity and divergence of TEs in the maize genome can be used to infer domestication history. For chapter 2, I'll look at patterns of TE content between populations in natural teosinte populations and explore the eco-evo drivers of that variation. And for chapter 3, I'll compare more distantly related perennial teosinte, Zea diploperennis, to explore the effect of genome structure and mating system on TE content. For this presentation, I'll almost exclusively focus on my first chapter, and I'll briefly go over future directions that will tie in my second and third chapter. But general big picture, each chapter shares developed pipelines for estimating family-level TE abundance and genome size from short reads, and each chapter relies on the same rationale, that patterns of TE variation and diversity on the population level in Zea are evolutionarily significant and have influenced selection and population structure in this genus. So it wouldn't be Pop Bio without some populations, and these are mine. I have a total of 180 range-wide samples across the 3 teosинtes in maize, and I have population level sampling with about 5 to 10 individuals per population for about 30 of those sites. The red dots are trial data sets that I've been using to refine my methods, and the purple polygon is the Balsas River Valley, which is the theorized origin of maize. In addition to my genomic data, I have a library of 27,000 TE families from previous work by Michelle Stitzer, Pop Bio alum. So as Jeff alluded to, I had a lot of trouble annotating TEs. Given that I have an extensive TE library and short reads for many different populations, how do I get to TE family abundance and diversity? There are many, many, many annotation software out there, but I had several restricting criteria. First off, whatever program I use must be highly computationally efficient. I plan on running this on hundreds of genomes, and I hope to graduate, so it has to be quick. Additionally, I, of course, want accurate annotations, specifically accurate annotations from short reads, which is notoriously difficult. And whatever program I use must be capable of parsing the complexity of the maize genome, which is highly repetitive and quite large. So I tried many programs, none of which met all of these criteria. Even programs made to annotate TEs from short reads in maize underperform. In bioinformatics and genomics in general, you often face a trade-off between speed, ease of use, and accuracy of your programs. But even the best short-read TE annotators made for maize either take a million years or have extreme false negative rates. What I have had success with thus far is co-opting a program called SourMash, which is a K-mer-based metagenomic tool, and using that to annotate my TEs. So SourMash, K-mer-based metagenomic analysis tool. K-mer just refers to a length of DNA that is K base pairs long. It's often used in genomics when using base pairs or entire reads is infeasible or inaccurate for whatever reason. The K-mers I use are 51 base pairs long, which means I chunk up my genomes in my TE library into chunks of 51 base pairs. But why use a metagenomic tool to annotate TEs? We thought, theoretically, TE families in a genome are comparable to microbial genomes in a metagenome. So what I do in this workflow is I generate K-mers from my genomes in my TE libraries and look at the overlap in K-mers from my TE library to the total K-mers in my genome. This will give you a general proportion that each TE family contributes to that query genome. To verify that this method works, that it was theoretically and quantitatively equivalent to TE proportion, I compared SourMash TE proportion to the real TE family proportion, which was estimated using the highest quality TE annotators from long reads. TE proportion from this workflow was on average about 97.95% accurate for this line of maize. However, if we look at specific TE superfamilies, we see that it's quite accurate for class 1 retrotransposons, which make up the majority of the maize genome. Ideally, I would compare actual base pairs that each TE family contributes to the genome rather than proportion, but in order to do that, I need to know genome size and coverage of my input files. There are K-mer-based approaches for this, but most are not accurate for maize given its highly repetitive genome. I've been attempting to estimate coverage in genome size using K-mer overlap at single copy conserved genes. How well does that work? Not well, at least not yet. To test this workflow, this idea, I pulled 6 different sequencing runs of the same inbred line of maize, B73, should have the exact same genome size. The only thing that's different is who made that genome, who sequenced that genome. And I trialed it at 6 different coverage amounts from 1x to 95x. I also trialed several different single copy conserved gene libraries to estimate genome size. These are just the highest performing 3 libraries that I had. So the real genome size is the orange line, and the calculated genome size for these 6 genomes is shown in the colored lines. For the genome at 1x, which is actually the reference genome, no overlap in coverage. Each base pair we know to be sequenced at 1x. All of my libraries were quite accurate, somewhere around 98%. But when I run my workflow on the other genomes, sequenced by other people for varying coverage, we get results all over the place. And the pink gradient shows the actual range that genomes in this genus can vary. As you can see, the error in these methods far surpasses actual variation in genome size. We were suspicious, though. Of course, this could be a bioinformatics issue, but the fact there's no consistent bias, over or underestimation of genome size, was a little bit suspicious to us, especially since this is an inbred line of maize. Genome size does not vary. So this raised some red flags, and we're now thinking perhaps this shows sequencing or extraction prep bias. What do I mean by that? It's possible that what we're seeing is due to uneven sequencing coverage between genic and non-genic regions of the genome. So between genes and not genes. Like I said, the maize genome is highly repetitive, majority TE content. And in previous work in maize, we see that differences in TE abundance estimated by short reads strongly correlates with who made that library. It's possible that different library prep could lead to an over or under representation of TE content when compared to genic content. Maybe TEs replicate from single strand more efficiently during the extraction procedure. Maybe polymerases have higher affinity for TE content at different temperatures or in different methods. Or perhaps repeat content anneals more efficiently during PCR because it's easier to find a matching pair when you're replicating. Honestly, all of this is mainly conjecture, but it makes sense that if I'm estimating genome size from genes, and I have a highly TE-heavy genome, and if one of these cases is true, it would lead to a biased result. More work needs to be done to verify this, but please take this as a word of caution. If you're working with short reads and you have a highly repetitive genome, you can't necessarily trust abundance to be proportional to actual genome abundance. So while this part of my pipeline is still in the works, let's move on to some actual research questions. What can I do with the data that I have? We know that TEs can be population specific and that TE content is highly variable between even closely related populations. Let's try to use TE content to estimate population structure. First comment that I often get when I say this is, why not use SNPs, single nucleotide polymorphisms? SNPs and maize are often shared between maize and the teosintase given their close evolutionary history. I generally expect that patterns in TE content will agree with SNP data, but TE variation may show more fine-scale resolution on the recent past than SNPs, and may be more informative for inferring maize evolution since it happened relatively recently. In my first chapter, I'll be exploring the diversity and divergence of TEs in the maize genome and how that can be used to infer domestication history, specifically through comparisons between maize and the teosinte it was primarily domesticated from, Parviglumis. As I mentioned in the intro, our idea of where maize domestication occurred and when it happened is still unrefined. And it's complicated due to continual gene flow between maize and the teosintase. In this chapter, I'll be attempting to use patterns of TE variation to explore this gap in our understanding of domestication history. This figure shows potential results where when we look at the whole genome, TE content across the genome, some populations of Parviglumis have more similar content to maize than other populations, potentially implying that those populations were the ones that maize was domesticated from. In terms of actual data, I've analyzed about 120 individual Parviglumis and maize genomes using my TE proportion workflow that I talked about before. We see on a PCA that populations of Parviglumis and maize separate, and that there's some population structure within Parviglumis, shown in the rainbow colors. Additionally, some populations are more or less similar to maize. Cool, let's look at a map. Specifically, the two populations that are most similar to maize, in the blue and yellow, these two populations are geographically closest to the Balsas River Valley, where we theorize that maize domestication began. Just a reminder, I've only run my workflow on my trial populations shown in red, but I have all the data for all the gray circles. Once I analyze more genomes across a larger geographical region with more resolution in the specific area, perhaps I'll get a few more candidate populations and we'll be able to narrow down the geography a little bit more. But besides relatedness, besides shared evolutionary history, what else might drive TE content? What else might describe these patterns that we're seeing in TE content? Maybe inbreeding and effective population size. Across several plant taxa, there's been a correlation with increased inbreeding and increased TE content. And generally, we can expect that with increased inbreeding and decreased NE, we should also see decreased TE content in a similar way to decreased genetic diversity. So do we see these patterns in our population? No. So plotted are the actual values for my trial population. TE content was not strongly correlated with effective population size, nor was it correlated strongly with genetic diversity or inbreeding. So if it's not demography that drives TE variation between populations, what else might it be? Maybe it's forces that affect the whole genome, such as eco-evo history, like elevation, environment, or maybe it's domestication history. Or if these factors do not drive TE content, does variation in the characteristics of the TEs in those genomes explain more of the variance in TE content between populations? All of these factors have the potential to drive patterns of TE content variation between population, but require fine-scale geographic sampling and genome-wide TE abundance to quantitatively explore. To hear me talk more about this, you'll have to come to my exit seminar. And to wrap up this presentation, wow, I'm speaking much faster than I did in my practices. Sorry. And to wrap up this presentation, I'll give a brief overview on what I'll be working on for my third chapter. Up until now, we have talked about the genome as a static landscape where T's can bounce around and it's just the T's in the genome that cause these variations. But that's actually an oversimplification of the genome. The chromatin landscape of a genome shapes T dynamics over time, since only sequence in open chromatin can actually be expressed. Previous work in maize has shown that open chromatin regions comprise of only about 2% of the maize genome, but are strongly associated with gene expression and recombination. T's in open chromatin have the potential to greatly affect genome fitness and function and should be strongly selected against. However, methylation or silencing of the TEs in open chromatin regions can negatively affect expression of nearby genes, which could also negatively affect fitness. This evolutionary tug of war is especially strong in the maize genome, which has many more TEs than genes, and only a few genomic niches where TEs and genes can both be co-expressed. So I plan on comparing TE content in open chromatin regions in maize and the teosynthase using something called ATAC-seq or ATAC sequencing, where only sequence in open chromatin regions is extracted and sequenced. And then I compare that to whole genome data that I have. So let's look at some data. I ran whole genome data and ATAC-seq data through my TE proportion workflow. This plot shows the proportion of TE families per superfamily that are present in open chromatin regions. Open chromatin is shown in orange and the whole genome is in green. So if there's no orange in the plot, that means none of those families are found in open chromatin. So we see there are no knobs or centromeric repeats found in open chromatin sequence. Great, that's exactly what we expect. They are not there. But we do see a chunk of DNA transposon families are found in open chromatin, and even more retrotransposon families are. But chromatin landscape can vary between tissue type. This is just data from stem tissue, which is vegetative tissue. What if we look at endosperm tissue? Endosperm is like the fleshy part in the seed, and we know that during endosperm development, T families, epigenetic control of T families is relaxed. So when we look at endosperm tissue, we see a higher proportion of all TE families are found in open chromatin, which is consistent with that theory. This data that I've used is from B73, an inbred line of maize that we use as the reference genome. And I mainly did it as proof of concept that this method works in identifying TE families. But I look forward to exploring population level variation in TE content in open chromatin, especially in the teosynthase, which I'm now growing a bunch of Diploparenus in the growth chambers, and we're going to do ATAC-seq, and it's going to be so much fun. But to summarize, For my dissertation, I plan on exploring how TE family abundance and diversity varies on different scales and for different life histories and demographies, between different genomic structures, and for different eco-evo histories. All this within the genus Zea. From this work, I hope to describe general patterns of TE content in natural and domesticated populations and explore the drivers of that variation. And a really quick acknowledgement for the people who's made this work possible. Big thanks to the whole Rossi Bar Lab, especially Jeff, Regina, Bebe, and Michelle Stitzer, whose extensive data I use. Huge shout out to Pop Bio as a whole, specifically to my third year cohort, and to my fellow Seed Grant besties, James and Katie. And thank you to NSF for the funding over the past few years. And a huge thank you to my lovely partner who had to listen to me practice this a million times last night. And now I will take any questions.
I'm fascinated by the endosperm. Great talk, by the way. Could you say a little bit more about why you would expect selection to be relaxed in the endosperm? Why would you expect to see them running rampant in the endosperm?
Yeah, well, let's go back 2 slides. There we go. So I'm not a developmental biologist, disclaimer, but endosperm development is particularly interesting from a TE perspective because of that relax of epigenetic control of TEs. And so developmentally, I'm going to hope to not say anything wrong here, but endosperm is actually triploid in diploid plants. So there's a mixture of male and female genetic material in there. And it's believed that epigenetic control of TEs is relaxed during endosperm development as kind of like a preparation of the immune system of the developing plant. So by letting TEs proliferate in the endosperm, you develop a lot of siRNAs, which are then transferred to the developing embryo in the plant, which can then have immunity to those TEs as it develops. So by allowing things to go crazy and then documenting what goes crazy and then transferring all that information to the developing plant, you start off with a more prepped immune system to TE proliferation. So you have prepped epigenetic control as the plant develops. And so it makes sense that we'd expect greater TE content in the endosperm and perhaps more TE families than in vegetative tissue would be active in this tissue type specifically.
So has anyone looked at the fitness consequences for like early seedling development?
That's a little tricky. I don't know that answer directly, but I know that in a lot of methylation deficient lines in maize, they've tried to look at this, but they don't often survive because you need methylation in a genome that has such high TE content like maize. I think they've done it in other taxa, but I'm not too sure.
Thanks. Great.
I have an oddly specific question. On your sour mash slide where you're showing the alignment of those different groups and how well that works, it looked like the worst were heliotrons. And I was wondering why that might be so notably different from the other groups.
Yeah.
Yeah.
So, funny story because we have time. When I started, Jeff said, you can do whatever you want so long as you don't touch helitrons. They're notoriously tricky to work with. Even in the maize genome, which we have all these resources, you would expect we should be able to explore helitrons, but they're really hard. Helitrons are also a little bit of an exception in the TE world. The way they replicate, it's like they're flat in the genome and then they excise and form this rolling circle where the ends can join and it forms like a loop. But in that process, it's like the ends have sticky tape on them and they pick up fragments of DNA. And so annotating these helitrons is really tricky because you also have random chunks of DNA inserted in the middle of your TE sequence. It's hard to know the length you're looking for. It's hard to know specific sequence you're looking for. especially when helitrons insert in each other and then pick the whole thing up as they move. It's just really tricky. And so, yeah, this method's not very accurate for helitrons, but also no method is. Unfortunately, we're not quite there yet, but yeah, great question.
Cool, thank you.
Any thoughts on the differences in the between stem and endosperm between the different superfamilies?
There's some theory showing about where these superfamilies tend to insert, whether it's in genic regions preferably, whether TEs prefer to insert in other TEs. Let me go back a million slides. There we go. So just a reminder, these are TE superfamily characteristics in the maize genome, specifically proportion inserted in another TE versus distance from genes. We assume that TEs that are closer to genes would be more likely to be an open chromatin, just based on where they are. Specifically, differences between endosperm and vegetative tissue, stem tissue. I don't have any great inferences, but potentially TEs that are mainly controlled by methylation would more likely to proliferate. Maybe TEs that are silenced by other TEs, or maybe TE superfamilies that are more sensitive to these tissue changes would be more likely to be expressed. Yes.
Great talk. I'm just curious if you think of Zia as having kind of an unusual TE story compared to other plants. I feel like I hear about monocots having, in general, having like unusually large genomes. I'm curious if you think that these dynamics are sort of happening in other systems.
Great question. So generally across, this is average trends across all plant taxa, genome size is directly proportional to TE content. So the smaller genome you have, the less TE content that you'll have. I think maize as a whole is a more representative natural genome across plants. As of right now, a lot of genetic studies, a lot of genomics work describing these patterns is done in Arabidopsis, which has an incredibly small genome and lower TE content as a result. Or maybe it's reversed, lower TE content leads to a smaller genome. So I would say maize is not an exception, and things we learn about maize using TE content is more likely to apply to natural systems than other plants. Specifically between taxon like ferns that have massive genomes and ones that have smaller genomes, I would say that it's all dependent on evolutionary history, some of which has to do with TE content, some of which has to do to polyploidy or anything else. So I guess generally I would consider maize to be a normal plant, but of course there's differences within that. Yes.
Hey, great talk, Natasha. Would you mind going to that last slide that you showed, please? That a quick question
This one?
I think the one
This one.
after this, yes. So I see in that kind of last tab there that one of the kind of differences that you're interested in is this clonal versus sexual kind of trends in TE kind of accumulation between there. I know there's been a little body of work that's been done in that kind of area, but just in regard to your system specifically, would you expect there to be more rapid accumulation of TEs in clonal versus sexual lineages because of the lack of recombination, or would you potentially expect those clonal lineages to have developed more effective mechanisms for potentially reducing TE load? Because I know there's some empirical evidence that suggests at least some relatively ancient asexual lineages do have less than predicted TE loads as compared to closely related like sexual ancestors. And so it's kind of like an odd story that I think where the empirical evidence doesn't really line up super well with the theoretical predictions. But regarding this system specifically, do you have any kind of like expectation about what you might find if that's a comparison you're interested in?
Yeah, absolutely. I actually have a slide for that. Let me find it. Just close your eyes. There we go. So I think a lot of the work that's been done on this is in Arabidopsis. Arabidopsis thaliana being a selfer, and its close relative Arabidopsis lyrata being an outcrosser. So you're definitely right. There's been conflict with theory about TE content and abundance between selfing and outcrossing. Selfing lineages often have lower TE content than expected. So generally, when we think of TEs, you can think of them as deleterious, so equivalent to genetic load. And when you have reduced efficacy of selection, which is often the case in selfing populations, we'd expect TEs to go crazy. Proliferate like mad, fill the whole genome, reduce fitness, etc. But that's not exactly what we see. And so this has to do, we think, with a balance between TE fitness and host fitness. If you produce a really unfit genome, the plant dies, the TE lineage dies as well, you can think of it. So TE fitness, in this case, in the case where there's low recombination, low selection, and low control of TE content, would mean, I lost my train of thought, but it benefits the TE to anthropomorphize a little bit, to reduce your activity so that the genome is more fit as a whole, because TEs contribute to the genome fitness. And so we see this in reduced activity in TE content in selfers, but we also see this on the genome side. There's stronger control of TEs in selfing populations than there are outcrossing populations. So stronger epigenetic control, more methylation, more tight chromatin structure, etc. So that's why we think there is less TE content in selfers and outcrossers. And I expect that same phenomena to be true in maize and its wild relatives, all of which have some level of selfing, some level of outcrossing. We know for maize what this is, especially since we breed maize, and so we can control that selfing outcrossing rate. This is not as well known for some of the other teosynthase, but we have general estimates. Yeah, great question.
No, thanks, that answers it really well.
Can you tell us about the cool vessels on your last slide, what they are?
Yes. Okay, so chicha is a fermented corn beverage that has cultural and social ties in the Americas for thousands of years. I actually passed around called maize morado, which is primarily used for chicha. Yeah, it has this really nice purple color. And so chicha is like this fermented purple drink, really refreshing, involved in religious ceremonies, cultural ties. And because it was so important for so many people, people often made chicha vessels. These were carved bottle gourds that often represented important things in your everyday life. Usually it was the gods, sometimes it was natural phenomena that you're involved with. And in this case, they're all rodents eating corn. These vessels would span several thousand years across different regions in the Americas. And so it is evidence that pest pressure in storing your corn has been an issue for a very, very long time. A large enough issue to make a whole chicha vessel that you drink chicha out of every single day. Yeah, great question. Thank you. I was hoping someone would ask that. All right.
Thank Natasha again.